Thank you very much. So I'm Fransisca from Houston Methodist Research Institute, and today I'll be presenting one of my projects on targeting the preterm labor management. And I will start with the technology itself, and then the second part I'll give an update about our process on progress in preparing for preclinical and clinical study. So as a bit of introduction, as we know from the last few days, and you all are probably working with nanomedicine, and I have a schematic here from a nice review in Nature Nanotechnology. It's about a range of application for nanomedicine. We use different materials for different targeting and different diseases. However, it's still missing the obstetric and gynecology. This is a very underestimated field, I think, and I was fortunate enough to be involved in this study. And my PI, Dr. Biana Godin, has started this study with OB-GYN, which, because there are not a lot of people who are working in this field, and OB-GYN itself are very, what do you say, like very old-fashioned. The technology has been there, like the medicine that has been used has been there for like decades. So there are a lot of challenges actually in this field. They have a lot of problems, and in this study we are targeting the preterm labor, and mostly to make sure that it increases safety and of the medicine that we are delivering, so that they are not reaching the fetus. There are a lot of other things too that we're involved in. Preeclampsia diagnosis as one of them, as well as the diagnostic and therapy. But for today, I'm just going to limit our presentation to the preterm labor. So the significance of preterm labor is very costly, actually. I'm from the US, where the healthcare is not the best, and well, it's actually really good, but it costs a lot. It costs like $59,000 per preterm birth, and then it costs $26 billion per year, caused by the prematurity itself, the mortality, as well as the following health problems that is caused by the prematurity. And indomethacin is one of the most used tocolytics. However, they can also cross the placental barrier from maternal to the fetus, and it causes a lot of problems. One of the main problems is the ductus arteriosus stricture, which make the artery that goes from the pulmonary system to the heart closed. So this is one of the main problems that has been that we have in the preterm labor. So as we know, like most of the free drug can transfer from the mother to the fetus freely. And we can change this depending on whether we want to treat the fetus or if we want to treat the mother, or as in our case, we want to target the placenta. We can use different kind of particles, and in our study, we did the research on the previous literature, and we found that liposomal formulation seems to be able to restrict the distribution to the mother side or to the uterus side and reduce the placenta transfer. So if we can restrict the biodistribution only to the mother side, it can prolong the administration of the drug and also reduce the morbidity associated with a preterm birth. So one of the main points that we want to have in this project is to have increased safety, especially to the fetus. And another one is that we also have increased efficacy, as we are not only adding the liposome indomethacin, but we also add the Autosemen. This is actually a drug by itself. It is approved in Europe by EMA, but it's not approved in FDA in America. They actually also tocolytic, which reduce the contraction. However, in FDA didn't approve it just because the efficacy is not high enough for them, not because of the safety concern. So we aim to use this and then conjugate it with the indomethacin liposome. And this is the schematic of our system. So we'll have indomethacin between the lipid bilayers, and we have a linker and then add the Autosemen. And for the size, we have around 154 and really high indomethacin loading efficiency because of their trapped in between the layers, and the zeta potential about -20. And we found that the lipindora, as we call them, are accumulated mostly in the uterus, and we found very low amount in the fetus. As we can see here, we have an increase of double of the concentration of lipindora compared to indomethacin, while in the fetus we actually have lower concentration of lipindora as we wanted. And the effect on the contractility, we checked the uterine contraction, and with the cell line, it did increase with, we use a mice model, CD1 mice model with LPS induction. Once they are inducted, there has an increase in uterine contraction, while lipindora actually reduced the contraction itself. This is also like the comparison in the number, in the percentage. And other than that, the indomethacin also reduced the prostaglandin concentration. We checked it, and it actually also reduces the PGE concentration. So as a summary for the technology itself, we managed to increase the indomethacin concentration with the liposomal lipindora, as well as reduce the fetus concentration of it, reducing the exposure to the indomethacin. And here is the uterine contraction. As you see, the lipindora caused like really lower contraction. And in effect, we actually reduce it up to like almost half of them actually have reduced in the preterm birth. So the second part is about the translation into the preclinical and clinical study. So we did the liposome in a normal way before, with using the thin layer formation with rotovapor and then the extrusion, which takes the whole day, like basically for 5, 6, 7 hours to finish. So what we're thinking is we can use the nanoassembler, the microfluidic-based liposome production, to simplify the whole system, because as we heard from the last few days, that it's hard to make the liposome as easy as possible, to make it easier to go through the to the FDA. And so we did like the whole month, we tried different settings, different flow rate, different ratio of aqueous and organic phase, and we found that actually our indomethacin caused some problems with the microfluidics. So according to the manufacturers side, we only have to do like washing steps, and those washing steps were not actually sufficient to remove the indomethacin. So for the longest time, we only get like liposome size 60 nanometers. So we figure out after we change the chip, we always get the better results. So we found that with this ratio, 1:1, 4 ml per minute, we get like really good size, about 182, which is a bit larger than the traditionally made particles. There are like around 150, and it has similar zeta potential. And the next part is we did like a whole system for the Cryo Protection during the lyophilization study. So we tested 7 different sugars as a cryoprotectants, and we found that the sorbitol seems to have like a better potential. And we found that 3% sorbitol has pretty nice size and pretty low PDI, and we decided to use that as our future. So we are going to also plan the GLP study in non-human primates for the PD-PK study in non-pregnant monkeys, as well as GLP studies in rats for single and multiple dose study. So as a summary, we figure out that this field is a really untapped field that we can have a lot to be done. And we have successfully done use Lipindora to target pregnant uterus and reduce the preterm birth. And now we're moving to the next phase of preclinical and clinical studies, and we use a nanoassembler and 3% sorbitol for lyophilization. And we are now have a lot of ongoing works on like establishing SOPs, as you guys probably know, as a lot of regulatory systems that we have to be done, like batch records for vialing, lyophilization, as well as the QC. So lastly, I would like to thank my PI, Dr. Biana Godin. Tracy, she's been working in the lab doing all the cryoprotectant study, as well as Translational Affair Office and GMP Core in Nanomedicine, as well as the OB-GYN side, who did most of the animal studies and planning on the clinical stuff. So thank you very much.
Thank you very much for this very interesting talk. So we have time for some questions from the audience. There's one at the back.
Thanks. I also noted that the field of gynecology or birth giving is really ancient. Do you have an idea why industry sheds away from novel products in that field?
Yeah, so a lot of OB-GYN that I talk to, they are very traditional, and it's not like in the cancer field where everybody's doing nanotechnology, and also not a lot of funding as well, I think. Probably that's one of the reasons. But yeah, I'm not sure why is it. Maybe certain personality goes to that part. I have no idea. But like, yeah, the indomethacin, they've been using it since the 70s, and they haven't changed it. So it's, but I think it's been changing. Like, actually, in the last, within the last few, can I get? The last few years, actually, when we started this one. So we published our paper, and then within that year, there are also 2 other papers that's coming out in this field. It's in infancy, but still it's growing, so hopefully it's getting bigger. Yeah.
I have one question for you.
Yeah.
So what is the pharmacological reasons for what you see? Is it because your liposomes bind to the cell surface and releasing or doing something at the cell surface, or they actually internalized by the cells?
What we're thinking is that the size matters, because the small molecules, they just go past the cells. Like they have the epithelium that goes to the fetus, and it seems like with the size, we increase the barrier and kind of reduce the passage. We haven't checked yet how exactly, because it is a really complex model. So we haven't checked yet how exactly the cell-cell transfer
because
looks
oxytocin receptor is expressed by the trophoblast
Yeah.
in the placenta, right?
Yeah, yeah.
But is that an internalizing receptor when you have actually attached to the liposomes?
Yeah, the point is we want to want to have it internalized by the placenta, but not going through the barrier. So that's
No, I mean the particle won't go through
Yeah.
the trophoblast. I mean that is the work done by Harish Patel in mid-80s. It's an old field, but then
I don't know why it's because there's a lot of problem with like pregnant women. They cannot take any medicine. That's actually a big market for it.
Right.
I don't know why it's like.
Well, we are glad you are doing this, so that's
Yeah.
great.
Yeah.
So any other questions? So that is the end of